Fuck it, she keep calling every single night. And I don't mind pushing through the line. I'm lost to the God, I'm in my mind. Swear I wanna die, yeah. When you cross my I wanna die, yeah. No, I'm not alright, yeah. I might start a
Assalamualaikum and a very good day everyone. My name is Muhammad Akmal bin Muhammad Radi. My matrix number is AS13418. Today, me and my group, which is group 5, will present about an evolution of Lycophyte vascular tissue. Okay, before we move deeper into the topics, let me give a little bit of introduction about vascular tissue. As we know, today, vascular plants, which is also known as tracheophytes, has dominates the natural landscape in nearly all terrestrial habitat. Vascular plants can achieve great height because they have roots that supports them, and with the roots, they also can absorb water from the soil, and they also have a vascular tissue called xylem that transport water to the stem to the leaf, and phloem, a conductive tissue which is transport nutrient in a plant. Further, the cell walls of conductive cell in xylem contain lignin, which is a material like candle that strengthens a plant cell walls. Thus, the evolution of xylem was essential to the evolution of upright and taller plant as we all can see today everywhere. Okay, now let me move into the origin of vascular plant. The fossil record indicates that the first vascular plant, such as Cooksonia, were more likely bushes than trees. They are not tall than our trees today. Cooksonia is Rhinophytes, a group of vascular plants that flourished during the Silurian period, which a very long time ago. More about 443.7 until 416 million years ago. Cooksonia are now extinct. The Rhinophytes were only about 6.5 cm tall and had no roots or leaf. I repeat, they had no roots and leaf. They are also more short than our plants today. They consist simply of a stem that fork evenly to produce branches ending in sporangia. The branchings of Cooksonia were significant for two reasons. First, instead of the single spirogyte in bryophytes, this plant produced many sporangia. Because they are produce many sporangia, they have more spores than others. Second, branching is a characteristic of plants that have vascular tissue. The sporangia of Cooksonia produce a wine-blown spores, which classifies it as seedless vascular plant. Like the rest of Lycophyte and Petridiophyte, we'll discuss in the next session. In addition, the Lycophyte and all other vascular plants have a dominant sporophyte generation.
Well, hello. My name is Elena and I will give you a short brief on Lycophytes. Okay, nowadays include around 1,275 species of Lycophytes that exist in three main groups. Okay. These three main groups is first one is called as crown pines or club mosses or Lycopodium, and second one is pipe mosses, or we call it as Selaginella, and the third one is quillworts or Isoetes. Okay. As you notice, all of these Lycophytes have moss in their name. Well, they are not actually moss because they have a different characteristic, which is they have vascular tissues. There is also one part of Lycophytes that is called as rhizome or the underground roots. Well, these underground roots is where roots develop from, of course, and branch from it. Okay. Then there is also one part of Lycophytes that is called as sporangia, and these sporangia are on strobili, and this sporangia is a spore producing structure. Okay. Some Lycophytes and some plants produce one type of spore that grows into one type of gematophytes. This type are called as homospores. Okay, contrary from homospores, heterospores produce two types of spores. First one is called as microspore, which grow into a male gematophyte, and the other one is megaspores that grow into a female gematophyte. I think that's all. Thank you.
Thank you, Akmal. So, hi, I am Muhammad Daniel from Kuliah 11, matrix number AS13329. So, I will continue about the unique structural adaptation that found in Lycophyte. So, the first unique structure that can found in Lycophyte is strombili. Strombili is a cone-shaped structure that we can found at the top of Lycophyte such as club mosses. Strombili, there is sporangia that can be found on the strombili. Sporangia, it produces spore, and the meiosis occur where the meiosis takes place and the spore are produced. And sporangia, also known as described as spore production structure. Then, the unique structure adaptation that can be found in Lycophyte is microphyll. Microphyll is the smaller size of leaf that have only one strand of vascular tissue. It differs with megaphyll, which is the size the leaf is bigger and it have many branches of the vein. We back to microphyll. Microphyll, it involves a simple side extension of the stem. Then, for example, club mosses. Club mosses, we can see that there is no branch vein on the leaf, and also the leaf gap between the leaf and the stem, we cannot see it clearly. It is like combined with the stem. It differs with megaphyll, where megaphyll, the gap is bigger than mesophyll. For example, megaphyll, if you look the leaf from the stem, the vein will come first and it follow up by the leaf. So that vein will be the gap between the leaf and the stem. While mesophyll, we cannot see it clearly. It likely combined with stem because the gap is really small. So, in overall, there are three unique structural adaptation that can be found in Lycophyte, which is first, it has strombili, second, it has microphyll, and the last one is it has rhizome. And that is all from us. Bye. See you later.